And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless. For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
Are they the same kind? Well, they have the subspecies, Canis lupus familiaris, which is the dog. Then there's the species, which does include the dog and the wolf. Man has been trying since he's been around, trying to classify animals and to put them in certain categories. For instance, does it have hair on the outside of its body? Well, then let's put it in a category. We try to do the same thing with tools and nuts and bolts and screws. We try to categorize them into different areas. I think that's just a natural human instinct to want to do that. Is our system correct? I don't know. They change things all the time. They that really ought to go in this species over here. Where does a platypus go? I don't know. I mentioned we organize in our shop our screwdrivers in one place, our pliers in another, and our hammers in another. But yet when you get a fencing pliers, which doubles as a hammer, a wire cutter, a bolt cutter, and a crowbar, where do you put that fence pliers at? I don't know, but put it somewhere. I don't think the fence pliers cares where we put it, and I don't think the dog cares where we think it goes. I don't think the wolf cares either. But we've decided to develop a system of classification. Kingdom, phylum, class, order, family, genus, species, developed by Carolus Linnaeus. And it holds up pretty good most of the time. You mentioned about a fox. Does a fox fit into the dog kind? Well, people argue about that. It's certainly a canine, so it's considered in a canine family. But is it in the same? Are all the carnivores canines? Well, no. The cat is in the carnivore family. So where exactly does the biblical kind fit in? Which is it? Is it genus, family, order? In some cases, it might be genus. In some cases, it might be family. Other cases, it might be order. I don't think the animals care where we put them. I think we're trying very hard to divide them, but I think in most of the cases, a 5-year-old can tell you if they're the same kind of animal, and a 5-year-old can tell you a pine tree and an elephant are not the same kind of life form. Okay? So Mr. Nelson, I saw you with your picture of you with him there, thinks that all life forms are related because they're eukaryotes. Well, that's the domain, eukaryotes, which includes flowers. Do you think flowers, let's take roses as an example, are roses related to dogs because they're eukaryotes, because they have a cell which has a nucleus wrapped in a membrane? Well, in that case, I could say sailboats are related to cars and bicycles because they have metallic nuts and bolts and screws. Well, that proves they're related. This is the kind of stupid logic you guys resort to. Of course they're related. They're both eukaryotes. That was Mr. Nelson's response. You've got to be kidding. That's an answer.
This video is about how ancient fossilized trackways prove Noah's flood to be true. You see, fossil footprints are a common thing when we look at the rock layers. However, there are major discrepancies in these tracks, which can help us easily falsify evolution and help us prove the biblical flood occurred. Trackways consistently appear in rock layers below the remains and teeth and shells and skeletons of the animals that made them. If you think that the Earth is billions of years old, then layers of rock are supposed to be representing millions of years of history. How is it possible then that so many footprints could appear millions of years before any physical evidence of the hard parts of the exact same creature that made them? If there are footprints, there has to be a creature. That's obvious. But where are their fossils? They're not there. How could millions of years pass with millions of creatures dying and none of them leaving a trace of their existence behind besides their footprints? Here is that solution. The global flood easily accounts for them finding trackways below body fossils because those creatures that made their trackways either fled to higher ground to survive or were lifted up and carried higher, their bones and teeth and shells eventually being buried by the rising floodwater. As a result, their body fossils would be found consistently higher than their trackways, and this is exactly what we see. We see the less mobile aquatic life buried by rising mud and sediments brought in by the tsunamis with larger and mobile creatures at the top, not because they evolved that way. And this pattern we see everywhere worldwide. It's a pattern found in trilobites, amphibians, and dinosaurs alike. Even according to the older paradigm, tracks are being found millions of years before body fossils and are just considered a curiosity. Modern-day bear and horse tracks in 300-million-year-old basement rock have been discovered. How many years are we talking about on average for these things? 10 million years for trilobite tracks and trilobite fossils. 35 million years between amphibian tracks and amphibian fossils. 10 million years between dinosaur tracks and dinosaur fossils. 245 million years between horse tracks and the first horse fossils. And 270 million years between the first bear tracks and the first bear fossils. That is more than a curious pattern to ignore. Ask yourself, how is it possible that in all of these millions of years, thousands upon thousands of bones and teeth and shells didn't fossilize? After all, footprints in wet mud are much easier to get rid of than the hard parts themselves. And if you look at the fossil record as a whole, there are tons more bones and teeth and shells than there are footprints. This is strong supporting evidence for the global flood, and nothing about it makes any evolutionary sense in their model. The pattern makes perfect sense from the perspective of a worldwide marine catastrophe, and it's a great example of how the data in the fossil record actually makes more sense when interpreted from the history recorded in Genesis. Here are more controversial tracks in slabs of a sandstone located at the Dinosaur Museum in Texas. There are layers of rock in the Grand Canyon area known as the Coconino Sandstone. Lots of amphibian tracks have been discovered over the years that are all visible tracks showing that the creature was climbing up a sand dune, and the tracks showing them trying to move in one direction only. But the angle and the shape of these amphibian tracks was a bit curious. It appeared that the feet were pointed in one direction while the angle they were walking in a significantly different direction. It was as if they were being pushed or pulled in a different direction than they were actually wanting to go. These tracks had been used to argue that the Coconino Sandstone was evidence of an enormous desert with lots of dunes and small amphibious climbing creatures. And as many scientists have observed, if you have a huge desert in the middle of the Grand Canyon, it certainly wouldn't have been subject to a global flood. Paleontologist Dr. Leonard Brand did extensive laboratory research with living amphibians and reptiles to see what they did in different settings. He observed what kind of tracks they made in dry sand, then he observed the type of tracks they made in water while walking on wet sand. He even added current to the water to see what would happen if they were pushed in a direction opposed to the way that they were going. The results were fascinating. He reported that the experimental trackways, which best matched those found in the Coconino Sandstone, were the ones made underwater while struggling against a current. It is the best explanation for these curious trackways, and it fits the biblical model to a T. Another piece of evidence, we have what was called a dinosaur stampede of fossil footprints in Australia, presumed to have been made on land. The tracks were recently reinterpreted to have been made in water, with many of the tracks being made by swimming dinosaurs. The report came out in the January 8th Journal of Vertebrate Paleontology, documenting a fossil site in Western Australia, where many of the tracks are nothing more than scratch marks from what was obviously a dinosaur, being buoyed up by the water, clawing at the dirt as it swam along. This is one of many tracks found there and around the world. But all dinosaur tracks have water somewhat involved. Every single one of the dinosaur track sites were made in what best can be described as tidal flats. Jenny McGuire and Deborah Mickelson of Colorado University at Boulder presented a paper at the 2005 GSA documenting what appeared to be a trail of dinosaur tracks from a dinosaur walking and getting buoyed up by the water, more and more losing contact with the seafloor. However, the drawing that they depicted is not the reality of what happened whatsoever. This is more evolutionist storytelling and blatant lies to your face. The fossil footprints are in the middle of a series of rock layers. According to Steno's stratigraphic principles laid out in the 1600s and still adhered to today, it would be assumed that those layers were laid down horizontally. This wasn't some seashore. The principles of initial horizontality, strata either perpendicular to the horizon or inclined to the horizon, were at one time parallel to the horizon, and nobody disagrees with this. Therefore, these tracks were not made by some dinosaur walking into the sea, but rather the dinosaur was on flat ground in what is called a tidal flat. And as the dinosaur was happily walking along, the water came in and buoyed up the dinosaur until it lost contact with the ground. This is powerful evidence that Noah's flood was true, not some dinosaur as they portray walking into the sea, especially in light of the fact that other dinosaur tracks were found in that area that were not swimming, meaning they outran the floodwaters in that area, as where this one did not. Dr. Kurt Wise was studying fossils in Death Valley, one of the few places on Earth where the Cambrian deposits are visible. He was looking for trilobites in just one of the lower areas, and so he got down on his hands and knees to investigate. But he found only a handful of animal burrows or pipes. These are made by organisms that once burrowed into the ocean floor. He noticed that the trilobite trackways moved higher and higher through rock layers. However, he found more curious burrows, eventually by the thousands. Further up, instead of finding burrows, he found complete tracks of the critters scurrying across the sediment. At first, he found only a handful of these tracks, but he found much more the higher he went. Suddenly, out of nowhere, he came across a mass graveyard of billions of trilobite shells. It was incredible. Kurt explained that scientists have found the same sequence all over the world. Burrows in one layer, trackways in layers above, and then the actual body of the fossil in the layer above the two. It's a curious pattern that is puzzling to anyone who thinks that the Earth is very old. Kurt provides a simple explanation by saying, as layer upon layer of mud swept over the trilobites, they struggled to dig out of their new tomb. As they ran across layer, they had just covered them and made new tracks. But as each consecutive layer piled on top of them, they eventually collapsed in exhaustion and died by the billions. Kurt's research is an excellent example of how creation science can look at the data that has been reported and provided worldwide and give a better explanation than the rescuing devices that evolutionists give.
Hey everybody, it's Standing for Truth. As you know, we've debunked a lot of evolutionists on this channel. We've also addressed and refuted all the top evolutionist arguments. Today, we are debunking Herman Mays, and it's going to be a lot of fun. Enjoy, guys.
They showed how common ancestry predicts a hierarchical structure between variant sites. So that's what you would predict based on common ancestry, is these variant sites that vary, should vary hierarchically. They should be groups within groups, basically. Single ancestry for each family predicts that sites will have no hierarchy and vary independent of one another. So there's a statistical test they do to see how hierarchical the data set actually is. And here's the difference between those data. So here the black data is the simulated data if creation was true, if there were separate ancestries for each lineage, and then the gray part's the actual data. The p-value associated with this difference is essentially 0, meaning there's virtually no chance there's a worse fit between the actual data and the expectations based on single ancestry. So again, you can rule out single ancestry. So here we go. Here's common ancestry or common design. Again, we have two models, a common creation event versus common ancestry. Here we have a whale, a cow, a manatee, and a shark. Okay, so here's our two models that we have here. Common design, you might be able to invoke a common design thing, but you would expect that common design would place the whale, the shark, and the manatee on the same branch because they're all designed for the same lifestyle. They live in water. Whereas common ancestry might not place animals with similar lifestyles on the same branch necessarily, because they can have different common ancestors. That common design tree is not what we see in the data, and we do see a common ancestry data there.
Dr. Herman Mays here attempts to argue that these nested hierarchies found in DNA, the same would also prove true for anatomical nested hierarchies, any form of nested hierarchies for that matter. Now, he argues that these hierarchies are only evidence for evolution and universal common ancestry, and this alone. This is where his mistake lies. He is asserting that function cannot be used to explain them. He rejects created heterozygosity and therefore sees no other way to explain the hierarchies. In fact, his statement of non-function is an assumption, not an empirical fact. There exists an abundant amount of literature that suggests that these third codon positions are indeed functional. And as a general rule, we can then predict function for these third positions, depending on which species were compared, whether they're the part of the same kind or not, etc. Now, hierarchies in general, they can be found at all levels in both the creation model and the evolution model. This evidence of nested hierarchies are agnostic to the debate. We can even find hierarchies on all levels in the design. Sedans and hatchbacks, of course, or sedans and SUVs will share hierarchies, since these modes of transportation are all incredibly similar, but we could even incorporate something as distant to a sedan as, say, an airplane. This example can be compared to, say, humans and birds, which according to our model are different kinds. Even these very different animals will share hierarchies, just like the sedans and the airplanes. When we focus our attention to materials used, for example, the types of metals, or we could even examine the types of tires used on both airplanes and sedans, what do we discover? We discover nested hierarchies. Therefore, these hierarchies are characteristics of design and not ancestry. I mean, think about it. Humans, pigs, rats, and ducks can share nested hierarchies. For example, they are all land-dwelling. Even though, according to our model, they are separate kinds, but since they are all land-dwelling and share many amount of other features, they can share similar nested hierarchies or design. As I've stated before, nested hierarchies in DNA, which is exactly what Dr. Mays is looking at here, are also predicted by creationists based on the design model. Now, because we look to function as a tool to differentiate between the evolutionary model and the creation model when it comes to DNA level nested hierarchies, Dr. Mays shows here just how superficial he can be in his approach to the creation model. Now, he suggests that our model, based on design, would predict certain animals to fall in the same grouping. Here, Dr. Mays believes that the creation model predicts whales and sharks should be part of one biological grouping, and then land mammals as part of another biological grouping. Now, this is because, as we know, whales are classified as mammals and not fish. Therefore, Dr. Mays thought that this refutes and falsifies our biblical creation model. But Dr. Nathaniel Jensen, in his rebuttal to Herman Mays, puts it perfectly. Dr. Nathaniel Jensen states, Linnaeus grouped whales with mammals, not fish. As a creationist, why would Linnaeus do this? Linnaeus based his classification on function. Superficially, since whales and fish live in the water, you might be tempted to group them together. However, despite their superficial outward similarity, they strongly diverge at the physiological levels. As I've said at the beginning of this part, Dr. Mays has taken a very superficial approach to our model. When it comes back to function, just to cover and go over the fact that we understand so very little of the DNA language. And much of Dr. Mays' approach and much of his arguments has to do with neutral variation or junk DNA, for example. But the fact is, junk DNA has been completely overturned, and we understand very little of the DNA language. So much of the claims made by evolutionists in regards to DNA and DNA function and junk DNA has been overturned. Pseudogenes, for example, they're now known to be functional DNA elements and not mistakes. Endogenous retroviruses have been overturned. They are now known to have functional roles, for example, in the placenta and in the embryo, among other important roles. And then they try and say that these roles and these functions have been co-opted, nothing more than a rescue device with no real evidence, no real testable predictions. We have really just begun to discover just how amazingly functional the human genome is. Just to think about how many experiments would actually need to be done to conclude with any real level of surety that a particular set of, say, DNA sequences actually has no function. And we know that we have preliminary biochemical evidence for function based on ENCODE. And this also goes for pseudogenes and a lot of the markers that they may use to build their phylogenetic trees. Yes, this evidence is preliminary, but it leads to future testable predictions on DNA function. The trajectory is clear. What the evidence suggests is genome-wide functionality, and this destroys Dr. Mays' central argument, because if we can prove that the vast majority of DNA differences are created functional DNA differences, this becomes a common design argument, as creationists have always predicted, and not an argument for universal common ancestry. If we both predict nested hierarchies, then we need to look to something else to differentiate between the models, and that is DNA function. And increasing levels of DNA function is exactly what we see.
The reason I'm bringing up the book is because how you answer that question deals with a lot of the topics that I bring up. So are all differences the result of mutation? Are some of those differences the result of created diversity? We're talking about very different processes, and it gets a much more complex discussion. We're talking about coalescence, and it's going to be difficult to not talk past each other unless we're talking about hypotheses that I'm actually proposing.
Now that we've debunked his claim on nested hierarchies on a DNA level, let's focus on his other claims. But first, we need to explain and go over what is the created heterozygosity hypothesis. Well, an important question to ask is, where does genetic diversity come from? Now, the major differences between our view as creationists and, say, somebody like Dr. Herman May's view, who holds to the position of universal common ancestry, is that we explain the vast majority of nuclear DNA differences not by mutation, as Herman May's would explain them by, but as pre-existing or created functional DNA differences. This created heterozygosity is a genetic difference between the models that has massive implications for the timing as to the origin of species. And that's what this debate is about. Those evolutionists that want to point to fossils, a bone found in the dirt, or a rock in radioisotope dating, for example, fossils, rocks, bones found in the dirt are not inherited sperm and egg. Traits are, genetics are. And that's where the battle is won. Now, if you are trying to explain the origin of species like Herman May's here just by mutations, yes, of course, you need millions of years for all these differences to accumulate. If the differences are there from the start, though, the simple operation of processes Genetic processes such as recombination and gene conversion can produce a visible variety in a single generation. This is essentially the created heterozygosity hypothesis. It makes testable predictions, and it is a hypothesis that has incredible evidence in support of.
So any coalescent time that you have for mitochondria that predicts that things go back, say, 6,000 years, you're going to have not just one, but you'll have actually a Poisson distribution of coalescent times with a big long tail like this that's older than the coalescent time for mitochondrial DNA. So all the genes, whether they're mitochondrial or nuclear, have different coalescent times. And
If you
for
assume that
mitochondrial
all
loci,
the
the
differences
coalescent
are the
time
result
is
of
much more.
mutations.
It has nothing to do with mutation at all. It has.
That's
it
how
has
the
to
differences
do.
are produced.
It has to do, because you're counting backwards in time. It has to do mainly with the effective population size. So just like here, I never in this example said anything about mutation. But clearly you can see the chance that if you go back one generation, if it's 4, is greater than if it's 10. So that means the time that you can trace back 2 sequences to a single coalescent is going to be more for the one with the bigger population size than with the smaller.
Oh yeah.
And that's true regardless of the mutation rate. And so that's not something you talk about. And there's a whole plethora of that's just the very, very basic undergrad version of this.
And I agree with all that. That's
every
what
gene
covers
every
the
gene is going to
textbook.
have every gene is going to have an independent history associated with it. And so you get all these genealogies that you're going to use to determine what the species history is, and it's going to be different. So if you trace back a mitochondrial coalescent time to 6,000 years, then that only means that there's a nuclear time that's much, much older than that waiting for it in the background. So
implicit in this theory is the fact that these differences arise by mutation, and that's a major difference between my explanation for nuclear DNA and the prevailing mainstream one. And I'm quite familiar with that.
They don't arise through mutation?
Isn't it funny how one of Dr. Nathaniel Jensen's main points in replacing Darwin has to do with created heterozygosity, and that the majority of nuclear DNA differences are actually created as functional DNA differences? Dr. Jensen also claims that although nuclear DNA differences are created, the mitochondrial DNA differences are the result of mutations. And this is why Dr. Nathaniel Jensen predicts mitochondrial DNA mutations should only go back 6,000 years to Adam and Eve. And these predictions and retrodictions have come true and continue to lead to future testable predictions. Now, it's quite obvious if you understand this model that if the nuclear DNA differences are created, and we know there are millions of them, then obviously if we were to do coalescence calculations, then it would appear those DNA differences resulted millions of years ago if they were the result of mutation. This is not what us as creationists, and specifically Dr. Nathaniel Jensen in his book Replacing Darwin, claims. And what's funny is that, as you can see here, Dr. Mays used that argument in the debate numerous times as one of his main arguments that he thought proved the creation model wrong. But all it did was proved that he either just skimmed through the book or didn't read the book. I think Dr. Nathaniel Jensen puts it perfectly when he states the fact that he advanced an argument such as this posits a view of creation in conflict with 3 chapters of my book, he says. That reveals he probably didn't read it. Now, to summarize, Dr. Mays said that mitochondrial DNA and nuclear DNA have different coalescent times. Of course, this is standard coalescent theory. Now, this claim made by Mays only works if we assume that all mitochondrial DNA and all nuclear DNA differences are the result of mutation. Now, Replacing Darwin Chapter 7 shows clearly that the biblical base model looks to mitochondrial DNA differences in 6,000 years in a way that leads to testable and falsifiable predictions. Dr. Jensen even wanted to test some of those with maize, and he refused. Now, if you skip to chapters 8 through 10 of Replacing Darwin, it is amazingly clear that this model explains the majority of nuclear DNA differences not by mutation, but by creation in the beginning, which leads to testable and falsifiable predictions. Dr. Mays can only attack a straw man of the biblical base model.
How would you say is my position on the origin of nuclear DNA?
The origin of nuclear DNA? Well, I think your
Nuclear
position
DNA
on the
differences.
origin of all DNA ultimately is God created it.
That's incorrect.
You don't think God created DNA?
What do you think I said in my book?
Well, you can explain it. I don't have it memorized. If I'm going to mischaracterize it, you can explain it right now. It's your chance.
That's why I want you to go through all the examples you gave. Just pick one. I want you to show me from my book. John Perry actually mentioned this last night. He's actually read what I've written, and he says he knows that one of the reasons I give for why so many people, 97% of the scientific community, disagrees with what I say. One of the things I say is I think so many people haven't read it, and I'd like to see some evidence from your slides that you've read the book. So I'd like you to pick one of the examples that you give for testing common ancestry versus separate ancestry. For the record, I actually talk, that's a whole other separate topic, but I'd like to see from my book, one of the tests you give, the falsification of common ancestry versus separate ancestry, how that separate ancestry prediction flows from what I've written. I make a lot of very specific statements, both in the book and especially in the endnotes, and I'm still waiting for you to point out one of those, and I'm disappointed you haven't been able to really give me more from the book and that directly addresses what I say. What I'd like to propose for the future is this, and we didn't really have time to discuss mitochondrial DNA. Here's a testable prediction that's in the book that we can both evaluate side by side. I think this is a pro-science way of taking the debate forward. What I say in the book is that if mitochondrial DNA differences are explicable over 6,000 years, well, the last 6,000 years, we all agree, is the history of civilization. So you should be able to see the stamp of this. The Roman Empire, the Greek Empire, Genghis Khan's empire, all of these things should be recognizable within our mitochondrial DNA differences, and not so much if mitochondrial DNA differences represent hundreds of thousands of years of history. One of the best test cases that will distinguish between evolution and creation is the transatlantic slave trade. And we've got 2,504 individual mitochondrial DNA sequences within the 1,000 Genomes Project that's freely available. Anyone can download, anyone can do this experiment. And I'm spent 2 years on this, close to being finished. I think we have a detectable signature. So why don't we do an experiment in parallel? You can, this is your expertise. You can download those 1,000 genome sequence, make a prediction.
Nathaniel, we just talked about this. For you to say that mitochondrial diversity only goes back 6,000 years, you're saying by necessity that the coalescent of the samples of mitochondrial DNA we have today can be traced back, so they all trace back, those mitochondrial lineages trace back to one within 6,000 years.
I'm taking the challenge to them now and saying, hey, let's go out and there's a fox in the woods. I bet you I can tell you how fast his mutation rate is.
The question.
It's the DNA is incredibly complex, but I'm trying to get you to see a bigger picture. Comparing similar DNA between me and all other Norwegians might prove there are genetic markers that all Norwegians have and all Namibians do not have. Okay, that's probably true, but we're still human and we're still interfertile.
I get that, Kent, but that's
you
not
don't get it. You do not get it. Humans produce humans.
You don't get it. DNA is complicated, and there's a lot we don't know, but this we do know.
Okay.
We can use neutral genetic variation to place your genome in the context of a particular population history. And the exact same methodology, using that same neutral variation, is the type of variation that I'm talking about when we look deeper into evolutionary history, differences between species or common ancestry between species and families, et cetera. It's the same technique.
I understand, heard every word, and understand. Now, so the similarities that I have with other Norwegians, even though I've never been to Norway, that same type of data you're going to use to prove that humans and grass are related if you go back far enough. Is that your? Is that
Neutral
your?
variation? Yes, and there's a reason for that, because it's the same process. It's the same process. We can look for patterns of common ancestry when we look at neutral genetic variation. Again, it's two models, and the data are going to support. They're going to support the predictions of one model or the other. Now, I haven't heard your model necessarily make any clear predictions.
Okay, I'll make one right now. I predict humans will always produce humans. No matter what climate you go to, no matter what country you go to, no matter how far back in history you go, you will never see an example, never, where a human has produced a non-human or come from a non-human.
Well, Ken,
that's my prediction.
I need predictions that we can test without the benefit of a time machine.
Go to the hospital. Go to the maternity ward. Go to the maternity ward.
Data we have right now.
Okay.
What predictions does your model make of the data when it comes to neutral genetic variation?
My model predicts humans will produce humans, and the mutations.
That's not what I'm asking. That's not what I'm asking.
No. You mean that's not what you want to hear?
No, that's not the question I'm asking.
Kent, it's not the question he's asking. Actually, he's asking you what you can do now to provide experimental data that supports your hypothesis of what you're trying to put forth for the young Earth creationist position.
I go to any maternity ward and watch when the baby's born. It will be a human.
But that's consistent with the evolutionary prediction as well.
That is consistent with the idea that humans and grass have a common ancestor? Come on. You've got to be kidding.
Why? Explain to me why.
Okay.
All of the fossils, you agree that there are geological strata, yes?
I agree that there are many layers of rock on the Earth. I do not agree they are different ages. I have right here. I have in our museum pictures of petrified trees standing up, connecting all these layers.
Now, call them whatever you want. I'm asking you a question. Call them whatever you want. In these layers, there are fossils. Yes,
I agree.
And they are organized in a pretty particular way. Yes.
The fossils are not always in that particular way. If there's any,
they are essentially always in that particular order.
Then you have been lied to, and you swallowed a lie.
No, I haven't. No, I haven't.
Okay.
Can you find for me a hominoid fossil in a very low layer next to a trilobite fossil? Can you find that?
That's very interesting. We have a section in our museum working on right now about trilobites living with humans all along. There might even be some.
Yeah, that sounds fascinating, though it's not based in reality.
Dave,
now you keep derailing me, so I never got to ask you my question. My question for you is that you believe that these layers were deposited during some kind of flood as waters were draining or something like that, right?
I agree. Yeah, the layers were deposited in a flood.
Okay, so then what I want to know is, number one.
Most of them, nearly all of them. There's still layers today being deposited, but I'd say 95% of what we see was deposited during Noah's flood. Yeah.
Number one, why do the layers have zero correlation with density? Why does the density not increase as you go down? And number two, why are these fossils in those specific layers, in that specific order, everywhere around the world?
Okay, they are not in that order, but let's take one question at a time. I have in front of me a sand art toy that has 4 different densities of sand: black, dark blue, light blue, and white. 4 different densities. But every time you flip it over, it makes 30 or 40 different layers.
Are any of those limestone or sandstone or granite? Are those in there?
Dave, I'm not sure what you know about physics or hydrologic sorting, but water automatically sorts things, especially during the tidal change during the flood in the days of Noah. The Earth is turning around, in spite of the flat earthers, and the tide goes up and down. And so if the Earth were turning for a year while the tide's going up and down with no interruptions, it would make thousands of layers, and the density would be completely confused. Generally, clams are found at the bottom.
What do you mean completely confused? What does that mean?
Well, during one tide, you would get more dense and light dense, and then 6 hours later, you get another tide coming in, bringing another layer on top of all that, so you may get more dense and less dense. Just Google stratigraphy, experiments in stratigraphy, stratigraphic experiments at Colorado, at the Navy.
So you're saying that tides exist everywhere on Earth, in every location on Earth at the same time. Is that basically what you're saying?
Well, during the flood in the days of Noah, the Earth was covered and the tides would cover the world. Now, some parts of the world have a high tide while other parts have a low tide. You see?
So how can you get the same strata everywhere on Earth? Tides occur on a coast.
If the earth were covered with water during Noah's flood, tides would occur everywhere, Dave.
What do you mean they would occur everywhere? Tides occur on a coast. Tides come in and out. If the world is covered by water, where is a tide? How does a tide?
Hold it. Let me explain, Dave. Here is the Pacific Ocean. They have a high tide and a low tide in the middle of the Pacific. You don't see it because you're in a ship floating up and down. But the Pacific Ocean actually comes up and down during the tide. What do you mean tide occurs at the coast? Have you ever studied anything about earth science?
I know about lunar tides. You're talking about the depositing of sediments in a particular way.
Yeah, right. See, the world is turning. At this latitude, 31.3366 degrees north latitude in Lenox, Alabama, we're turning 886 miles an hour toward the east. If the tide were going up and down during Noah's flood, the water would have to rush sideways at really high speeds to fill that bump, and then rush down the other way at high speeds to lessen the bump.
Really high speeds? What does that have anything? How is it any different than tides today?
Okay, it's not. It's not. But the tides today get interrupted by banging into the continents. Earth turns toward the east, so the tides generally hit the eastern coast a little more because the tide is being drawn out by the moon, lunar gravity.
You're trying to ascribe some kind of magical quality to tides that doesn't exist. We see what tides do today, and we see what they do.
It's very simple. Here, look at this. I just flipped this thing over. These 4 different densities of sand made, probably 20 different layers.
And again, are any of those things sandstone or limestone or granite or anything that would be applicable to this discussion whatsoever?
I think it's pretty obvious, even from the name. You might even get this. Sandstone is made from sand, okay? Sand is moved very easily when tides move back and forth. Water going back and forth.
But it's not the same material. How does it possibly compact into
it would have to harden.
It
would have to harden over the next few hundred years after the flood. After Noah's flood, the layers would harden, and some of it's a chemical reaction. There are different types of sedimentary rock.
And how does this process occur magically at every location on Earth at the same time, such that the strata are the same everywhere on Earth?
Well, first, if you think the strata are the same every place on Earth, you have really been lied to in your education.
Nope.
You're the one who doesn't have an education. The strata are all different areas. And all over the world, petrified trees are found standing up connecting these strata. So how
that's
can
not
you
a
claim
problem.
they're different ages?
Some are in volcanic ash. Some could be due to a local flood. That is not evidence against anything.
I agree. I got it. You need to watch my video number 6.
I don't need to watch any of your videos, Kent.
You didn't? I'm sorry, I didn't hear you. Did you?
I didn't, and I don't need to.
Oh, I know you didn't. It's obvious, but you need to. It would help you a lot, okay?
No,
you really do need to. Okay. Tell you what, I'll watch some of yours, and I'll put you on my list to whack an atheist. Are you an atheist or just an agnostic, or what are you?
I don't really feel like telling you because it's not really part of this discussion. It has nothing to do with God whatsoever, unless we want to actually talk about the Bible like I wanted to do earlier. Can we?
No, that wasn't the title of the debate. The debate was the evidence for evolution. You're claiming there is evidence for evolution.
No, the title of the debate is evolution versus creation, and the Bible is your evidence for creation.
Well, no, even without the Bible, I could say it's obvious there's a creator. I think somebody created this coffee cup. Had nothing to do with the Bible. This has a creator.
Yeah, another man-made object that has nothing to do with anything. Your argument is that a designed thing exists, therefore everything is designed. Well, I can tell you that something like a snowflake forms spontaneously, so everything forms spontaneously. Neither of those conclusions follow from the premise. They're both bad arguments. You should stop saying it, even though it's one of your favorite ridiculous things to say.
Well, it's not ridiculous. It's true. And you're not getting it, obviously. So I need to say it over and over again. I'll tell you what, most of the audience is getting it, even though you are not. It is true. Do you believe the first living cell, the first living creature on the planet, came from something that was non-living? Because I used a coffee cup, which is non-living. Do you believe the first living cell came from something non-living?
Kind of a gray area, but at some point you'd have to demarcate that. Yes.
Okay. So how did this non-living, whatever it was, soup, collection of chemicals, you can have whatever you want. How did
you're not saying rock anymore?
Well, that's what the textbook shows, that the rock.
Oh, no, it doesn't. But that's what you always do say. But it's interesting that you're not saying it right now because you know that
I'm going to try to get you to admit it, Dave. Now listen. Do you believe the Earth began as a hot ball of rock 4.625 billion years ago, or whatever number they're using now?
Yeah.
Okay. Did the rocks cool down, and then it rained on the rocks and produced chemical oceans containing chemical soup?
You can call it soup. I wouldn't, but fine.
Most of the textbooks call it a soup. Okay. Do you believe this soup somehow came alive?
Eventually,
soup
over
did
millions
not
of years.
come alive. Organic molecules organize themselves into ordered systems, which eventually we can describe as life.
The living things came out of this organic molecules came from this soup, from the rocks that got rained on. Dave, you believe you came from a rock. You just admitted it and don't even know it.
No, I don't. I believe that organic molecules produce life, but you know who does believe that humans came from rocks? You do. You believe that God created Adam from dust. So you're describing your ridiculous belief.
Now,
not mine.
Let me try to explain. I'll try to explain it simply. This coffee cup is made of 100% natural materials. Okay, ceramics from the ground and stuff like that. For a man to take the materials from the ground and produce a coffee cup is understandable. For an intelligent God to take the dust and make a human, that's logical. For the dust to make itself into a human, that's not logical, Dave.
All right, everybody, let's jump right into the story of Mitochondrial Eve and talk about the timeline from the past until present with all of the different information that they have given the public. But let's jump into the science first. Going back to 1996, we find that Howell discovered that looking at the control region of mitochondrial DNA, that there were 2 mutations in 81 transmission events. What this means in layman terms is that there are mutations which are transmitted to future generations, which can be observed and cataloged. This opened the eyes of the secular community that maybe their fossil record clock assumptions are wrong. These results placed a most recent common ancestor just thousands of years ago. The reason that they had all assumed that they would find a slow clock is because they all believe in deep time-scale evolution to be true, and invented a dating technique to validate this belief by calibrating a clock against the fossil record. By adding in an assumed ape-human split 6.5 million years ago, they were able to obtain mutation rates based in favor of evolutionism. A year later, a study was done, peer-reviewed and published, which again shocked the scientific community. It obtained the results, placing Mitochondrial Eve, the mother of all living humans, at just 6,500 years old. But this date was not good for evolutionists, for many obvious reasons. And they were very angry that a creationist is the one who discovered these numbers. So they came along a year later and retested using much more advanced methods and using a huge demographic, narrowing down the results. Unfortunately for them, down to 6,000 years exactly, proving the biblical timeline and a young Mitochondrial Eve. All of the results proved that their phylogenetic assumption method rates were nonsense and should be thrown out. They have proven a high observable substitution rates in human mitochondrial DNA, placing it at roughly 20 times higher than their estimated rates. These findings were so accurate they were even adopted by forensic analysis by the FBI and used today, so their accuracy is spot on, regardless of what evolutionists would like you to believe. Another large study using 2 different methods also came to this same conclusion. And that same year, a separate study confirmed it again. Now remember, one should detect around 1 mutation per 90 transmission events. MUM discovered 1 mutation per 59 transmission events, which is even faster. And Holland again the same year, looking at HVR1 and HVR2, discovered 10 mutations per 306 transmission events, again confirming young Earth creation. A few studies obtained few mutations, or none. This was because they either neglected to look at germline and somatic mutations, or they changed the way they define mutation rates. For example, here, we define the mutation rate as the rate at which mtDNA type of an individual changes, rather than at the rate of mutation accumulation in individual mitochondria. So again, it goes back to a biased way of thinking to obtain the results desired. As you can see it here in action, looking at Cavalier L's work in early 2000. He listed 0 mutations out of 292 transmission events. But this is because he only counted homeoplasmic nucleotide substitutions and also used estimated divergent rates as approximations. And even he admits in his conclusion that the pedigree rate of the control region is significantly higher than the phylogeny divergent rate assumption. So what happened is they wanted to figure out what's going on here with this. So what they did in 2003 is they took all the pedigree studies, or trio studies as they're now called, and they were put for accuracy using all possible effects, from homeoplastic changes, selection, random genetic drift, and more. The conclusion confirmed earlier results, even with the studies that showed zero mutation added in with this calculation, and that was that their phylogenetic assumption methods all failed, and young Earth creation rates were much more accurate. So taken into account that even by adding in biased studies, which gave them no inherited mutations in trio studies, they still helped confirm our suspicions. And in that same year, the Annals of Human Genetics confirmed young Earth creation mutation rates. A year later came testing in the form of mathematical models, using all data compiled and focusing on different approaches. Both results they obtained concluded that a recent common ancestor for all of mankind was just a few thousand years ago. Then again, in 2012, we were vindicated yet again. They discovered, looking at 15,585 human protein coding regions, that SNVs, known as single nucleotide variants, that the human population growth was recent at just 5,115 years ago, max. Not even close to a 200,000-year-old bottleneck, like they tell the public. So again, observable rates completely contradict the evolutionary assumption rates. Then, in 2012, it was discovered that nearly all near-neutral mutations and harmful deleterious gene variants arose in the last 5,000 years. They state the vast majority of these may have no phenotypic or functional consequences we yet know of, meaning he's mostly talking about near-neutral mutations handed down from generation to generation. In all, about 14% of all mutations identified were found to be harmful. The results are based on a genetic study of roughly 6,500 Americans of all demographics. The study's conclusions suggested that the human population acquired an abundance of single nucleotide genetic variants in a relatively short time. This is just another piece of evidence that is a disaster for the evolutionary theory, but not a problem whatsoever for us young Earth creationists. We have now mapped the entire genome of worms. We know everything there is to know about their genome. And after that, they looked at their mutation rates in hopes to confirm evolution and finally answer the mutation rate problem. Look at what they stated after discovering their mutation rates were just like humans and ticked 100 times faster than expected compared to the evolutionary assumption rates. As you can see, a complete embarrassment for their theory, and more validation for young Earth creation. And you can read on the bottom that they concluded this study by saying the higher mutation rate results have not been accepted by many scientists. Now you know why. And here is yet another observable genome study of human genetic variation obtaining young Earth creation numbers as well. This 2009 study, which even added and assumed archaeological dates of early primitive man to be true, they still obtained results that confirm young Earth creation. They directly state, these within human estimates are also higher than estimates generated from phylogeny-based human-chimpanzee comparisons. Our new estimates established a rapid decay in evolutionary mutation rates between approximately 2,500 and 50,000 years ago. Yes, you heard it from evolutionists themselves again. A 6,000-year biblical timeline would not be out of this range. They're the ones that have to resort to a bottleneck as a rescuing device to fight against the observable evidence, as you can read for yourself. So now you can see the assumed human and chimp ancestry rates and the non-assumed human and chimp ancestry rates. I'll let you determine which one you want to believe. Now, again, what is the actual evidence compared to the story they tell us? Well, we only have one single line of both male and female lineage with low genetic diversity in the world today, which could only come from a single recent human common ancestor. 2. There is no genetic bottleneck evidence even in their own geological column. 3. There is no actual genetic evidence for the story they wield about other human beings being alive prior to Eve. 4. There is no logical explanation why all these supposed 10,000 women living at the time of Mitochondrial Eve, or the thousands and hundreds of thousands that lived before her, all died off while leaving zero biological trace of their existence. 5. There is no logical explanation why all these supposed children of these women who supposedly existed alongside of Eve all perished without a trace either. 6. It's completely illogical to conclude that all these 9,999 other women who supposedly lived alongside of Eve all died without leaving a trace, or that their 9,999 other women only produced male children. And yes, that's a theory, and that is why we only see one direct maternal line today. There is no possible way that that scenario is true scientifically, mathematically, or even logically. 7. There is no proof they lived at different eras in time either. As a matter of fact, there are even studies that have been done that show that the two lived at exactly the same place at exactly the same time, contrary to what they thought, given their theory. So even secular science proves that there is evidence that they lived at the same time, at the same place. Literally every rescuing device they use to fight against the observed actual evidence is more ridiculous than an episode of South Park. It's sad that the people do not see it because of their bias. But look at what they say. The audacity is astounding. Even if all the data pointed to an intelligent designer, such a hypothesis is excluded from science because it is not naturalistic. And that is because of their assumption bias that they say that. We know that the out of Africa theory is made up, and that the humans did not arise from the L1 node deep in Africa. We were created with what is known as created heterozygosity, and this is easily explained with genetics, which I will prove. You see, the Bible says that man was created in the Middle East, specifically in the Fertile Crescent. Well, what do people look like who live there? Well, they have brown skin. Evolution says that man arose in deep Africa at the L1 node, which would have made them have black skin, just like all Africans. What does science tell us about skin colors? Well, skin color, or melanin, is governed by multiple genes, and genes come in two pairs. 2 from each parent are inherited, just like letters in the alphabet. Let's look at A and B right now. As genes code for large amounts of melanin, are capital letters A and B, as where lowercase a and b will represent low melanin in the skin, so pale white people. Therefore, black skin races can only carry capital letter genes, capital A, capital B, capital A, capital B. They can only ever produce more black skinned people, just like white people, which carry all recessive lowercase gene letters and can only ever produce low amounts of melanin children. So Adam and Eve had to have been brown-skinned people, therefore to be able to code all the different skin colors for the people that we see on the earth today. So even simple genetics proves the out of Africa theory is wrong, and proves that the biblical created heterozygosity model is true.
We can see here that all the evidence regarding mitochondrial DNA, Y chromosome, the low genetic diversity that we see all points to Adam and Eve. Evolutionists need to ask themselves, if they have an open mind and they were to look for evidence for Adam and Eve, what would they expect to find? Because all human beings are profoundly similar genetically, and this points directly to the out of Africa scenario due to the resulting inbreeding of a population bottleneck of 10,000. How is this even remotely feasible? A Y chromosome of apes and man, and how they're so dissimilar, less than 70%. The Y chromosome should be the most similar.
But you know what's interesting? When the Human Genome Project mapped the human genome, they said there's only one race of people. You know what that confirms? The Bible. Because we all go back to Adam and Eve, we're all one race.
That just makes the Bible consistent with nature on this one point.
Oh, well, that's good. I'm glad they agree on that.
How do you explain the fact that ERVs, or dead viruses, are in the same exact places in humans and chimps, but not other species?
So good question. Regarding ERVs, it comes down to the fact that are they really the result of ancient viruses? Because now we know that the evidence suggests that these endogenous retroviruses and these retrotransposons, for example, they're indeed functional, and not just functional, not just low levels of function, but these endogenous retroviruses, they have crucial functions. They help regulate genes and even determine cell types. So like I said earlier, the evolutionist now has to claim that they were co-opted, but that is just more fancy storytelling. So these are important DNA elements, just as we'd predict based on created genetic diversity.
Of course it does.
Neff, hold on, hold on a second.
The article.
Hold on.
Question, please. Can I ask a question? I'm not saying it does. Please, can I ask a question?
Why did you cite it? Then why did you cite it? It doesn't.
Because this is the very mechanism that supposedly created the 80% difference. Can I ask a question?
According to who? According to who? Not this paper.
Wait, you're saying it's not?
That's the question.
Okay, so let me ask you a couple of questions. Okay, are you telling me, is it not true that 80% of the proteins in the human
Can I ask a goddamn question? I do not normally get angry, but I am frustrated.
James, hold on. The difference.
This is not a conversation. This is a monologue.
James, the problem is this: the evolutionists, when you point out this kind of evidence, they go ballistic and atomic on you because they can't answer for it. And so that's what you're doing. Moving the goalpost endlessly. So I'm going to ask a couple of questions to get to the meat of it. And the first question is this: is it not true that 80% of the proteins in the human genome are different by one amino acid, right?
I cannot deal with. I am very angry. Let me tell you. Let me talk for 1 minute. Will you let me talk for 1 minute?
Is that not true,
guys?
Will you please let me talk for 1 minute? You keep on saying you're going to let me talk, and that you never do.
Is it not the same mechanism that created the difference in our proteins?
Oh my God! Please,
Shush!
The other people who have their microphones muted, will you please tell us, do you think that Nephilim keeps asking the same question over and over again and does not allow us to talk about it?
The problem is this.
Please tell us, do you think that Nephilim keeps asking the same question over and over again and does not allow us to talk about it?
The problem is this.
Well, here, I want to ask you a quick question that I asked in the opening. if there really was Adam and Eve, okay, we started from two people, okay, what would you expect to find in our genetics? You being an open-minded, skeptical individual, what would you expect to find?
Incest. I don't know. I think it's possible for any two individuals from species to create, to basically sustain that species and to multiply. I don't see, with the right dice rolling, I mean, you can basically marry your sister and have a perfectly fine kid. So, you know, which obviously happened, right, according to your story. But I don't see a problem with that.
Because once again,
I don't know what would happen.
Yeah, but I have to point out your assumption for the audience. You're assuming that we explain the vast majority of DNA differences as mutation. You know, when we accumulate 100 new mutations per person per generation, we have a lot of other inherited DNA differences that are being passed on that are not mutation related. So even the evolutionist actually resorts to and looks to pre-existing heterozygosity. Yes. We're just saying the original heterozygosity was engineered by God. That's also why we look to function, of course. But the question I asked you, you didn't really answer it honestly, I find, because I think you knew in your head, you probably thought, okay, I would expect to find low genetic diversity, evidence for one Y chromosomal ancestor, evidence for a mitochondrial DNA ancestor, and we can look at observed mutation rates, not accounting for evolutionary assumptions, and it takes us right back to 6,000 years. I mean, everybody's Y chromosome and everybody's mitochondrial DNA is incredibly similar. That's why the evolutionist looks to the population bottleneck. Even they believe in mitochondrial Eve and Y chromosome, Adam. Of course, they resort to unrealistic mutation rates and phylogenetic assumptions to come up with their date as to when they existed. But that is the evidence that one should expect to find if they're open-minded and a critical thinker like yourself. I mean, what other evidence would you want to find for me to convince you that, hey, listen, Adam and Eve exist, the Bible is true?
There's this, oh, where the hell was I going with that? Totally lost my train. It's really late for me.
I think you are much more simple-minded than you realize. Let me show you the picture here.
Oh, really?
This textbook shows, and I got all the textbooks beside me, that a human and a bird have a common ancestor, which was an amphibian. Let's see. Let me find a family tree that they show. Here, because I mentioned about the frog and the prince, okay? They have these textbooks with these charts all the time showing all these animals going back to a common ancestor. Do you believe you have a common ancestor with a strawberry, Dave?
Yes.
Okay, you're welcome to believe that. That's not science. That's a religion.
Yes, it is science.
Nobody's
it
ever seen a
absolutely is
strawberry,
science.
Dave, nobody's ever seen a strawberry produce a non-strawberry. You might get a bigger strawberry. Or
That's not
but it's
what
still a strawberry.
evolution says happens. Nothing in evolution ever says that dogs produce non-dogs. Nowhere is that said. What does that have to do with anything?
Okay, Dave, calm down. Let's take that statement. Nowhere does evolution say dogs produce non-dogs. Is an amoeba a dog?
No.
Just yes or no. Okay. Did the dog come from an amoeba?
What? They had a common ancestor.
Dave, come on, yes or no? Did the dog come from an amoeba?
What does this have to do with anything?
You're not
what does this
asking
have
a
to do
question,
with anything?
son.
It did not come from an amoeba. No, it did not come from an amoeba. What does that matter?
Well, the charts in the book show the amoeba turning into all the creatures, including the
dog.
dog.
No, they don't. Show me a book that shows that, Ken. No book shows that. Show me anything coming from an amoeba. Is an amoeba a eukaryote?
A eukaryote simply means the cell contains a nucleus that's bound in a membrane. And I'll use the same terminology you've been using. In your simple mind, you think because they're all eukaryotes, therefore they're all related. Couldn't that not be a common design? You know, all the cars on the highway have metal in them, iron.
You're just using the exact same analogy. Why are you talking about cars? They're not biological organisms. This is not a valid analogy. You're saying that there's something designed, therefore everything is designed.
No. Adding biological ability to reproduce makes it 1 trillion times more complicated, much harder for you.
They're eukaryotes, Kent. They're all
Did the
eukaryotes.
protista turn to a pine tree and a human? This chart says they did. This is what kids have to learn in school. You do believe a protista became a human and a pine tree.
Over 2 billion years.
Son?
Yeah.
Huh? Okay, just admit it. If you want to believe something that dumb, that's fine.
Okay.
But don't call it science. This
All
isn't
you're
science.
saying is that you don't get it, so it's not true. So let's get into this. Let's get into this. Kent, we're not getting anywhere with this. You're going to take all the animals forever. Let's talk about genetics. Let's talk about genetics.
What? Somewhere for the protista to become a human, new
So
information
you want to
has
answer
to
my
be
questions,
added.
and you're just going to go on and on and on and on about protists turning into whatever you want to talk about.
I want the audience to see what a dumb religion you guys have chosen to follow.
Not a religion.
It is a religion. You believe.
No, there's no moral code. There's no social structure. There's no
imagine. Here you go. Put your name on right here. Okay. Put your name on right here. You imagine that over millions, billions of years, the protista could turn to a pine tree and a human. That's what the charts show, and that's my objection. It'd be much simpler for a skateboard.
Do you know what a paternity test is? Do you know what a paternity test is?
Here we go. 202. Alt-DV 202. You asked for, here is a tree of life used in the textbooks that shows humans and arthropods and worms coming from a single cell, sponges. Do you believe we all came from sponges, Dave? Do you believe this textbook is right? You do, okay.
I forget the name of Jensen's book, but it's recent.
Replacing Darwin?
Yes.
Yeah. That is such a pile. Oh my God. It's really a bad book. I haven't read the book. It's really a bad book. Guilty of not reading the book. It's really a bad book. I haven't read the book. It's really a bad book. Guilty of not reading the book.